Good morning, everyone. I think everybody's back in. I saw a few of you defying the rule to leave the building, and I did the same to warm up. My name's Sherry Margulies, and I'm part of the organizing committee for today's event, as well as the co-chair of the Community Advisory Committee for the Canadian Cure Enterprise, which is CAN Cure. And I also work with the Canadian HIV Trials Network as their community engagement co-lead. I've been living with HIV for almost 30 years. And I was diagnosed in 1993 in the early days when we really had no hope, no treatment. And 3 months later, my infant son was also diagnosed with HIV. I'll be turning 60 next month. My son will be turning 30 this year. And he has a 2.5-year-old son as well. So we see that we've certainly prolonged life and quality of life for people living with HIV. It was interesting. We'd actually hoped to have my son come today, but he's too busy. He's working. And it would have been really nice to have him here today, but I had an opportunity to chat with him for quite some time before I came here to talk about what our different perspectives were on cure. And it was really, very interesting. And for my son, it was flat out. I don't ever want to take meds again. I don't ever want to hear the word HIV again. I don't ever want to have to disclose to a new partner ever again. I just want it over. And for him, it was quite absolute. A cure for him was never meds, full health, no more stigma, and no more anxiety when it came to new relationships, whether it be with friends or sexual partners. For me, the cure is a little different. Having lived 30 years of adult life as opposed to 30 years from infancy, I probably have not as high expectations of what a cure might look like. A cure might look like a 3-month or a 6-month or a once-a-year injection for me. And for many of us, being on antiretroviral medication and living a life without a viral load is almost like a cure. Unfortunately, we still, those of us who are long-term survivors, still carry the burden of being of taking experimental medications throughout the years, as well as other illnesses that affect many people at age 60, but more so the people living with HIV. And the biggest thing, I guess, that we'll probably never be able to cure, and what is probably the most difficult part of this whole equation, is that even when we cure HIV from a scientific point of view, we can't cure the stigma. We can't we can't go back and erase the memories of those of us who lived with HIV through very difficult times. We can't bring our friends back, which is really unfortunate. And I remember one of the first AIDS conferences I went to back in the early 90s in Vancouver. And someone came with a banner and a button that said, "All I want is a cure and my friends back." So unfortunately, we can't meet all of those goals, but today we're going to take an opportunity to meet with some of the professionals who are working across the life cycle. So from infancy until our older age. And our first speaker today, I have the honor of introducing, is Deborah Persaud. I hope I'm pronouncing your name right, Deborah. Good, good. Deborah is the co-program director at the PAVE, which is Pediatric Adolescents Viral Elimination. And it's a part of the Martin Delaney Collaboratory. And it's the first program of this collaboratory to focus on research for an HIV cure in children. Dr. Persaud is a professor in pediatrics in the Division of Infectious Diseases at Johns Hopkins University School of Medicine and the Department of Molecular Microbiology and Immunology in the Johns Hopkins Bloomberg School of Public Health. She's also the interim chief of the Division of Infectious Disease, and her expertise in HIV latency and pediatric infections. She's the chair of the HIV Cure Scientific Committee for the International Maternal Pediatric AIDS Clinical Trials Network. And I should take this off if I'm speaking, sorry, where she oversees the development of clinical trials toward HIV cure in children. Welcome, Dr. Prasad.
Good morning, and actually maybe afternoon, but thank you for sharing your story and the story of your son and highlighting that a child born with HIV or acquiring HIV can live to 30 years and 40 and 50 and can be a parent. And so congratulations on being a grandmother. And I'll start. Thank you, Corinne, for inviting me to be part of this session to give an update on clinical trials in HIV cure research. And so I'll start with, I've served on the scientific advisory board for Merck and Company. And so I thought I'd start with just a summary of where we are in 2022 with respect to the landscape of HIV cure trials and interventions. And this actually agenda started in 2012. So it's about 10 years old. And this is where we are. And you could see in the top four, which is what I'm going to focus on in my presentation, is really, if you look in the blue highlighted sections, is what we're trying to achieve for remission and cure in neonates. And it's really using the regimen that Maria described this morning from 2005 of early therapy in children, AZT and 3TC with Kaletra, and here we added nevirapine. And in this trial, these trials were actually really propelled by the single case of pediatric remission of 27 months in the Mississippi baby who received very early treatment at 30 hours of life. And so the overarching goal of these first four trials is really beginning testing, implementation of very early treatment strategies, really, and an impact P1115 to see if we could identify one case of remission. So as I present the data on P1115 and a follow-up trial, the Totelo trial from the early infant study in Botswana, I want you to kind of think about the regimen that we're testing in these studies. And then the second is that we've moved into our second stage trials, our combination with broadly neutralizing antibodies. So in P1115 in version 2, we now have advanced to using integrase inhibitors, which is raltegravir, for which we had to obtain PK in neonates before we could move this perhaps better regimen into neonatal treatment beyond nevirapine and Kaletra-based therapies with VRC01. And then in 2008, which is really not a curative trial, but looking at reducing reservoirs. And as Sharon said earlier, one strategy is reduce and control. And so that's the primary goal here. How can we get to early reduction in reservoir size towards control with immunotherapeutics? And I do want to point out an impact of P1107 study, which is where the New York patient came from. It was actually a study we planned for pediatric patients because most pediatric patients get cord blood transplantation. We didn't enroll a pediatric patient. We didn't enroll 2 adult patients. And this is from which the New York patient, the study from which the New York patient came from, that was led by Yvonne Bryson at UCLA. Just recently completed from Paula Palmer's group in the EPICAL Consortium, the Hurricane trial looking at long-term suppressed children and adolescents receiving a therapeutic vaccine. So it's an HIV DNA vaccine followed by 2 MVA boosts. There's no treatment interruption in that study. It's really looking at biomarkers of HIV persistence. And the last that's in development is 2039, an impact study looking at combining HIV conserved vaccines, Tom Hankey's vaccines, with and without broadly neutralizing antibodies that will include an ART interruption step to assess for sustained virologic control. But I do think it's important for us to start, I'm going to go very quickly now, is that these are actually the recent numbers as of today, 160,000 new infections in infants. So perinatal infections continue. There are 98,000 deaths. So we really need to improve our treatment strategies. 1.7 million children living with HIV. We know from the COVID pandemic that access to HIV testing, PMTC services, and diagnosis in women have been disrupted. And so we need to pay attention to really prompt diagnosis of infection during pregnancy. There's a treatment gap, and these are recent data. Only 52% of children are receiving ARV coverage globally compared to 76% of adults. And virologic failure is high. We're dealing with 40 to 50% rates of virologic failure, highlighting the need for this agenda to get started. The key features I think that gives us hope with perinatal infections is the opportunities to intervene early. We know when infants are exposed to HIV, and so we can act pretty early to reduce reservoirs. And as Sharon pointed out, the unique immune landscape, there's a high proportion of naive cells, up to 90%, so less susceptible target cells. And there's also an immune tolerant environment with which this is established. Now, this is a very busy slide, but I really want to highlight where we are 20 years later into early therapy of infants. And we know that early therapy, the reservoirs do become smaller over time. They start out with large infected cell concentrations in infancy, about 2 to 3 months of age, but these become very low over adolescence to less than 10 copies per million cells. And we've shown that there's paucity of intact induced proviruses in these long-term suppressed adolescents, a low viral diversity, a great landscape for immunotherapies. Recently, we've shown that the latent proviral reservoirs is harder to reactivate in children and adolescents living with HIV with long-term suppression, suggesting differences in states of latency and perhaps the properties of the cells in which the virus resides. And so altogether, I think these data from multiple groups, many present in the audience, support the unique aspects of HIV persistence with early therapy. So I kind of want to move to where the pediatric trials are, and just to point out that our trials are in resource constrained settings, low middle income settings where the epidemic lies. And as you can see from this slide, and there are more participants enrolled in investigator-initiated studies, but over 1,500 participants from 38 sites in 12 countries have enrolled into Cure trials. So we have many little Toms out there participating in these trials with their moms to get us to this point. So the first data set I'm going to present is Impact P1115, which is the very early treatment trial to assess HIV remission in infants to replicate the Mississippi baby. And quickly to go over the numbers, 2 cohorts of infants were enrolled. Cohort 1 were infants who were identified as high risk, meaning that their moms did not know they were infected. They were identified during labor and delivery. And so these infants were preemptively started on a 3-drug regimen, undergo testing, and then continued on study ART if they were infected. And so you can see in cohort 1, 440 mothers and infants were enrolled, of whom 36 infants were infected and 34 continued on study treatment. 2 participants withdrew prior to step 2. In cohort 2, we also wanted to see whether there was a window of opportunity to intervene to reduce reservoirs with very early treatment. And so these infants were started within the first 10 days of life, but had initiated a 3-drug prophylactic regimen within 48 hours of life. And so what you see on the left is version 1 regimen that's completed in the data I'll present, and it's really AZT3TC nevirapine with the addition of lopinavir ritonavir when age appropriate, and then de-escalation to lopinavir-based AZT and 3TC when virologic suppression was achieved. And version 2 now, as I said, now is studying raltegravir and VRC01. So we're actually getting safety and PK data of these 2 agents in the infant cohort now, and that's enrolling. In terms of infant characteristics, most of these infants were from sub-Saharan Africa, but about 30% of the infants in cohort 2 were from North and South America. We've identified that within this trial, multiple HIV subtypes are included, ranging from predominantly subtype C, but we also have AD recombinant AE, and subtype F. So giving us a good representation in terms of virologic responses to very early therapy to build upon. So this is a busy slide, but just to kind of highlight the timing, when you think about interventions and enrolling a mother-infant pair in a study, an intensive study with a lot of visits and evaluations, you can see the timing of ARV initiations and study enrollment for these infants. And so in cohort 1, the median age of ARV was about 7 hours, and in cohort 2, about 32 hours. So very early initiation of ARVs. Most of these infants were breastfed in cohort 1. And what I want to point out, if you look at these are infants that are being born with in utero infection, you could see plasma viral loads are quite high, 4.9 logs before any ARVs are started within the first 2 days of life, and 4.1 in cohort 2. And HIV infected cell concentrations by the standard singleplex DNA assays is high, 2.4 and 2.8 log-10 copy per million, but with good CD4 levels at start of therapy. I showed a slide to show the frequency of evaluations because each of these dots represent a visit, and this is just data through the first 2 years of life, where frequent plasma viral load and DNA loads were monitored, including HIV antibody tests. And we did come up with a biomarker profile to identify which infants would qualify for ART cessation and ART interruption. And these infants had to maintain strict virologic control, meaning absolutely no detectable RNA from 48 weeks onward, not even target detected below the limit of detection to stay on study and had to test HIV antibody negative and have normal CD4%. And so these are the summary data. And again, I want to remind you that this is with AZT3TC and nevirapine with the addition of Kaletra. And what we saw by 2 years of age in terms of estimated probability remaining free of virologic failure. So again, strict virologic suppression, about a third of the infants in cohort 1 and nearly half in cohort 2 met this strict virologic criteria with this minimal regimen of therapy to continue on study in consideration for ART cessation. And here the summary data of the biomarker profile at 2 years of age. So amongst these infants who got to this point at 2 years of age, you could see 83% in cohort 1 tested antibody negative and 100% in cohort 2. 64% in cohort 1 had non-detectable DNA by 2 years of age and will be followed continuously to assess for getting to non-detectable DNA, with 71% in cohort 2 and normal CD4% for age. So overall from this study, we estimated about 19% of cohort 1 would have met criteria for ART cessation by 2 years of age. Now with the COVID pandemic, we obviously couldn't do an ART interruption study. And so this part of the study is now begun and is ongoing. I want to point out that there are other studies of very early treatment, again, confirming that this is a scalable approach in multiple settings. And the slide on the left is from South Africa, the Leopard study, in which similarly infants were started on very early treatment, but here they're started within 7 days of life. And what's the endpoint here was really maintaining virologic suppression and normal CD4%. And at the end, I think they found that of only 19% of the infants at a year of age met the criteria for ART cessation, and so stopped the study because it felt it wasn't feasible to continue that part. But nevertheless, we gained information on implementation of the strategies. And then on the right, to highlight the study, the EITS study in Botswana with Roger Shapiro, with virology done by Matthias Lichterfeld, looking at the proviral composition and showing that amongst these 10 infants that they followed sequentially, a high proportion reached a low level of DNA of 5 copies per million by 2 years of age. And in this study, they identified innate immune responses correlated with intact proviral reservoir size. So from these, I think we have some key findings here, and that is that low reservoirs. I hope that what we've learned so far is that we can achieve low reservoir size in a subset of infants, meaning on non-detectable DNA by 2 years of age. Obviously, you know, the sample limitation, we need more cells to really get down to how low below 1 copy that level is. As I said before, we're in the process of ART cessation to assess whether we can detect 1 case of remission through the strategy. I think the important point of these studies is they've really propelled the field of HIV therapeutics in children, investment in therapeutics and investment in immunotherapeutics, and at the same time informing safety and implementation strategies and management in the space of infant HIV remission and cure strategies. I'm always very worried about time, so I might be racing, but I want to end with the adjunctive therapy, now moving along to using broadly neutralizing antibodies to achieve remission. And shown in blue is really a summary of broadly neutralizing antibodies that have been studied in infant populations for prevention and treatment, and the several studies within which this is being conducted. Impact 2008, which is one of the first studies to look at combining VRC01 with standard ART in infants around 2 to 3 months of age, will be presented during the late breaker session later in the week. And so I'm going to end with summary of the Tatello study led by Roger Shapiro. And in this study, what they did is from their cohort of very early treated infants, so treated within the first week of life, 48 hours to a week of life, they used a strategy of combining broadly neutralizing antibodies to maintain virologic suppression. And so the cohort were children 96 weeks of age or older enrolled in the study. They had to have had private plasma viral load less than 40 copies per mil for about 24 weeks prior to study. And here's the important part here. This is the first study combination broadly neutralizing antibodies in a pediatric population. The children were given IV infusions every 4 weeks of VRC01-LS and 10-1074 with the schedule as shown here. And so here are the essential, the outcomes or the steps for this study. There were 40 screened, but eventually 25 who received the combination of BNABs. And the plan here was to interrupt ART following BNAB administration, and then to follow viral loads through 24 weeks, and then restart therapy at the 24-week standpoint. And so among the 24 children who enrolled in the study and stopped ART, what they identified, 44% maintained plasma viral loads through 24 weeks of BNAB-only treatment. 56% had viral rebound, and that viral rebound occurred within 4 weeks of ART cessation. Importantly, it's the trial had a predefined criteria of 30% as a success, and so they exceeded their estimated percent success. So I would say this is a success in terms of very early treatment followed by a combination BNAB to maintain virologic suppression. And so overall, the study findings were the combination was safe and well tolerated. And importantly, all children resuppressed pretty quickly when we started on lopinavir-ritonavir-based ART. So together, I think these findings really provide support that combination BNAB, very early treatment, combination BNABs may afford sustained virologic suppression in the perinatal period or infancy. And so I hope I've convinced you today that despite the the kind of archaic nature of the regimens we're using to achieve these endpoints, we've actually achieved substantial progress in investment, development, and implementation of HIV Cure trials in infants and older children. There is a potential, I think it is possible for ART-free remission, very early ART. And it's really exciting now with raltegravir in place, and now with dolutegravir rollout, for which we have dosing as young as 4 weeks of age. And then the investment in provision of broadly neutralizing antibodies and combination antibodies to really help propel this field forward. So I think important progress have been made to date, and provides a framework for us for building future trials. And the nature of this session is across the age spectrum. And so our future therapies and therapeutic immunizations and BNABs will focus on the older age group. There are several pediatric viral reservoir presentations being presented at this meeting, highlighting, I think, the differences in reactivation potential of the reservoir, the effects of broadly neutralizing antibodies as part of primary therapy, an important study on the CNS reservoir in perinatal infected young adults with long-term suppression that we provided, and then some correlates of CD4 and viral load trajectories. So I'm going to stop here, but I do have to acknowledge the P1115 team, the Totello team, the Impact Network, and the support, and my laboratory team. And I'll stop there.
Thank you very much, Dr. Prasad. That was very interesting and exciting, particularly for someone who has tried to give medications to infants, and that was back in the day of ritonavir. So I'm sure liquid ritonavir, none of you ever want to take that. My family's Jewish, and when we have our Passover Seders every year, we say what our own, when we talk about the plagues that of the Jewish people that on Israel or on Egypt, we make our own plagues up. And when my son was 10 years old, he put his drop of wine on the plate and he said ritonavir. But with all that said, it saved his life. So, and got us to where we are today. So these arcane treatments, as you call them, were very important, certainly for him to have health. It's my pleasure to introduce to you today Dr. Jessica Prodger. She tried very, very, very hard to get here in person, but unfortunately multiple plane delays and cancellations prevented her from being here in person. However, she is going to join us virtually this morning. And she's the assistant professor and director of Global Health Systems Master's Program at Western University. That's here in Canada. Oh, then how come it was so hard for her to get here? And holds a Tier 2 Canada Research Chair and I got here from that area, genital immunology and STI prevention. She received her PhD from the University of Toronto and completed postdoctoral fellowships at the Johns Hopkins School of Public Health and at the Laboratory for Immunoregulation at the National Institutes of Health. Dr. Prodger is a longstanding research collaborator at the Rakai Health Sciences Program in Uganda. And through this collaboration, her research aims to increase inclusion of people living with HIV in sub-Saharan Africa in biomedical HIV research to ensure that the unique features of all populations affected by HIV are considered in development of a cure. So welcome, Jessica.
Thank you so much. Can you hear me okay? Thumbs up. I can't see anyone. Okay, I mean, I'm not seeing anything in the chat or the Q&A. I see myself on my
So
screen.
We see you, we hear you.
Great. Okay. I'm so sorry. I couldn't get out of the London, Ontario airport. I could have driven to Montreal for the amount of time I spent trying to get on a flight. And here I am. I just wanted to be there so badly in person in Montreal. So thank you so much for having me. Thank you so much to the organizers for pivoting and letting me present online. I'm really excited to be a part of this meeting today. The agenda is just awesome. I've loved the talk so far, and I'm looking forward to those in the afternoon. So the title of the talk, in my talk, is Considerations for Cis Women in HIV Cure Research. And it's true, the data that I'm presenting today is almost exclusively based on cis women participating in research. But I think a much more awkward but more accurate title would be Considerations of the Attributes of Biological Sex that are associated with cis women in HIV cure research. And the reason why I say that is because even though the data was generated in cis women, it doesn't only apply to cis women. So when we think about biological sex really does not fit very well into this sort of binary categories of female or male. But if you were to think about female biological sex and you were to think about what are the attributes associated with it, okay, so the first one, you have 2 X chromosomes, right? And cis women certainly have 2 X chromosomes, but so do some men. XXY males have 2 X chromosomes, and intersex people, trans men can have 2 X chromosomes, and other gender diverse people. Another attribute of female biological sex is a unique epigenetic profile. And part of this is driven by your chromosomal complement of sex chromosomes, but it can also be modified by exogenous hormones later in life that can influence gene methylation. And so this means that XY people or XXY people who are on feminizing hormones, like trans feminine individuals who may be biologically or chromosomally male, can have attributes of the epigenetic profile of that's associated with female sex that we may need to consider. And then finally, and this is where the group gets really broad, is female sex hormones, estrogen and progesterone, because people who are chromosomally male on feminizing hormones like trans feminine individuals would fall into this category. But then people who are chromosomally female and are taking masculizing hormones like transmasculine individuals would have their estrogen suppressed. So there's a lot of different parts to female biological sex, and they certainly don't only just apply to cis women. So please consider all of these groups throughout the course of this talk and when you are looking at data about female biological sex. Okay. So one of the first indications that we had that sex was important to HIV comes from HIV pathogenesis. So this graph on this slide is generated, it was published in a paper in 1999. So these are people who are not on ART as per the guidelines at that time. So they have a positive viral load. And what many different authors have noticed, or investigators have noticed, is that early in infection, females have a lower viral load than males do. However, over the course of time without treatment, the rate of increase of the viral load is higher in females and males, so that females progress to AIDS with about the same time frame, despite starting at a lower viral load. And what this means is they actually have a faster progression for a given starting viral load, which is interesting. Females also have higher CD4 T cell counts and higher CD4 to CD8 T cell ratios, both before infection and after treatment. And part of the reason for the believed reason for this discrepancy, both in early viral load and in rate of progression, has to do with immunological differences between the sexes. So there's a lot of important immune genes that are located on the X chromosome. And so you think, well, gene copy number wouldn't matter here because one of those two X chromosomes is inactivated, right? Well, it turns out that process is very ineffective, or at least there's a lot of errors in it. And about 20% of genes have biallelic expression for if you have two X chromosomes. So genes like TLR7, which can sense HIV RNA, it's the way immune cells can sense HIV, or one of the ways. FOXP3, which is an important transcription factor for T cell lineage commitment. It commits the cells to immunomodulatory cells. And 10% of all microRNAs are all on the X chromosome and might be affected by X chromosome number. In addition, epigenetic modifications are different in immune cells taken from males and females. The methylation patterns of those cells and the transcriptomes are different. And so what we think is happening here is that through these differences, females have higher interferon alpha production. So they have biallelic expression of TLR7, so they're better able to sense HIV. And then a higher level of estrogen is actually enhancing the downstream response to the TLR7, making more interferon alpha, which is a protein with direct antiviral activity. So you have lower HIV viral load early in infection, but then that also means you have more immune activation. So you can have faster progression to AIDS over time. But we haven't really had to worry about this in terms of treatment or progression because thankfully, thank you, antiretroviral therapy, most of them are targeting the virus. And so they're effective in both males and females. However, when we start to shift our focus towards cure, many of the curative agents that are under development right now are targeting host factors, and they're targeting immune pathways that we know to be different between the sexes, like TLR7 or PD1. And there also can be targeting epigenetic pathways and host genes that may be different. And so historically, we haven't had a lot of participants who are women or female in cure studies. In 2016, Curnow and co-authors published 11% of cure trial participants to date had been women. So we're not able to stratify in the results of these cure studies to try to understand these factors and how they might be different in male based on biological sex. So a good place to do that actually is in sub-Saharan Africa. And as mentioned, I'm a long-term collaborator at the Rakai Health Sciences Program in Uganda. And in Uganda, the HIV epidemic is generalized and it's predominantly heterosexual transmission. And so what that means is that males and females in the Rakai district in Uganda who are living with HIV are very well matched. They're very similar for other factors that may influence their HIV disease. So we enrolled 90 adults who were living with HIV, 57 of them were female, and all of them were ART suppressed. And we did two types of reservoir quantification in them. So one was just looking at all of the cells, quantifying all the cells that have any HIV DNA in them. And so a lot of that HIV DNA will be defective and can't make replication competent virus. And then we also did the QVOA outgrowth assay. So that's represented by this like little light blue circle. And what that relies on is our ability in vitro in the lab to be able to reactivate those cells and get them to make HIV. And what we know, at least certainly in subtype B, is that not all of those proviruses that are intact that could make HIV will get reactivated in the lab. We don't know why they don't reactivate. They don't make virus all the first time. So that QVOA is an underestimation. And it's an underestimation that may be biased, as I'll show you. So here's our data from that trial. And you can see here on the left that females have about a half log lower outgrowth in the QVOA assay than males. But when we look at their total HIV DNA, much of which is defective, it's about the same. In fact, the point estimate is even a little bit higher for females in this data. And I'm going to talk about the potential interpretations of that on the next slide, but I just wanted to make note that we also did have some other immune characteristics on most of these participants. And so we tried to look for correlates of reservoir size. So like what attributes or what other characteristics in the females or the males separately are associated with having a larger or smaller reservoir. And I just want to point out one, this data is published, so you can take a look at it in JCI Insight, but one of them that we found is that, as you might expect from the literature, showing that cells harboring intact HIV DNA are more likely to also be expressing PD-1 on their surface. We found that in our male participants, this positive correlation, so larger reservoir size if they have a higher abundance of PD-1 positive cells. But we didn't find this correlation in females. And in fact, other authors have published previously differences in expression PD-1 between males and females. So this is potentially important for immunologists if we're including PD-1 antagonists in HIV cure protocols. Okay, so back onto the interpretation of that reservoir size data. So back to that schematic. So we saw that for both the males and the females in our study, they have the same amount of HIV-1 DNA, but the QVO outgrowth was higher in the males than in the females. Okay, so what might this mean? Well, it might mean that females genuinely do have a smaller replication competent reservoir. Perhaps they have more hypermutation, more ApoBec activity, so that those are areas for future research. But it could also be that we were just getting really poor latency reversal in the QVO assay for our female participants. So they actually do have exactly the same size of replication competent reservoir as males. We just couldn't reactivate it. And there's a fair amount of data to support that this might have been the case. So in vivo observational studies have noted previously that the amount of HIV RNA levels in a person's blood, this is in the pretreatment era, varies with menses. So during the follicular stage, when estrogen is peaking, the viral load is coming down. And this has been backed up with a lot of in vitro data. So estrogen inhibits HIV-1 transcription through the estrogen receptor alpha, which can bind indirectly to the LTR of HIV and suppress its transcription. And in support of this, blocking the estrogen receptor using estrogen antagonists can release latency in an in vitro model of latency, and it can also augment the latency reversal seen with the HDAC inhibitor vorinostat, labeled SAHA here on this graph. And in addition, if you add estrogen in the form of beta estradiol to this system, you can limit the efficacy of that latency reversal and limit HIV expression. So these observations led to the clinical trial, the MOXIE trial, which represented the first cure research trial performed exclusively in women. It was successfully completed, and we learned a lot from the MOXIE trial in terms of willingness and feasibility for participation of females in HIV cure research. But the trial, which was using an estrogen antagonist to see if it could increase the efficacy of HDAC inhibitor vorinostat, saw no increase in no effect of the estrogen antagonist. Now, all of the participants in this trial were postmenopausal female, and actually only 4 out of 27 participants actually had a reduction in estrogen levels. So it's not to say that it wouldn't potentially work, but there's other considerations like toxicity of tamoxifen and reproductive aged women and things like that. So what do I think are the considerations moving forwards for those biological attributes of cis women, which may apply to many people for cure research? So I think a big one that we need to consider is estrogen may be limiting the efficacy of latency reversal agents in females. And it could be interfering with our QVOA assays. So I think, we need to think about that when interpreting the results of our studies or designing studies so as not to accidentally introduce noise and be able to understand what's happening in our female participants. In addition, female sex hormones and your sex chromosome complement may also impact immunomodulatory cures that are under development. So things like TLR7 agonists, maybe they might work better in females than males. That might be based on the hypothesis of the biallelic expression. But something like PD-1 inhibitors potentially could not work as well. So these are things that we would have to be looked into. In addition, we have the problem of subtype layered on top of sex. And so what I'm talking about here is in sub-Saharan Africa, where for HIV prevalence, the number of individuals infected with HIV who are female actually sees that of males. Subtype is different than in North America. So, and the problem is that most of the techniques that have been developed to quantify the reservoir and to be able to track the efficacy of cure interventions and trials have been developed for subtype B and aren't effective in places like Uganda that have, or haven't been validated in places like Uganda that have subtypes A and D, or in the rest of sub-Saharan Africa where subtype C is very prevalent. So, we clearly need more diversity in cure research to be able to start to understand these questions and address them. But this is going to need careful design. We need to start thinking about, for females who are cycling and are enrolled in research, where they are in their menstrual cycle. We need to think about puberty, pregnancy, menopause, hormonal contraception. We need to consider whether or not someone is taking transition-related hormone therapy. And when they enroll in our study, if they take women, what does that mean in terms of hormones and chromosomes? And I also think to be able to achieve this diversity in cure research, we need to have more community engagement. So, you know, as I showed you previously, historically, 11% of participants in cure research have been female. And this is partially provided the concept that females are hard to enroll in cure research. And the MOXIE study showed that it is feasible, but we do need to do work on community engagement to help generate a population of really well-informed people who can make decisions about participating, especially as novel agents with risk start to be tested. Okay, so I just want to acknowledge on this slide all the individuals from NIAID at the NIH and the Rakai Health Sciences Program, Hopkins and Western, who contributed to generating the data that I showed you today. And a huge thank you to the people of Rakai who are participating in that cohort. The cohort's now been running for 7 years. So we're going to have some really exciting longitudinal data, hopefully coming out for you soon. And then all of our funding sources. Thank you very much.
Thank you, Dr. Prodger, for this excellent presentation on considerations for cisgender women in cure-related research. Our next speaker is Caroline Dufour, who is PhD candidate in Dr. Nicolas Chemon's lab at the Centre de Recherche du Centre Hospitalier de l'Université de Montréal since 2018. She obtained a master's degree in molecular biology at Université du Québec à Trois-Rivières under the leadership of Dr. Lionel Berthoud. Her main research interests include understanding persistence mechanisms of HIV latently infected cells during long-acting antiretroviral treatments. Caroline?
Thank you for this great opportunity to present you a little bit of what our lab have worked on concerning end-of-life research. So just a glimpse of what everything can be done on various end-of-life research in the future. So at first, I want to make a brief overview of this presentation for the community members. This project has focused on understanding HIV persistent in deep tissues. This is of interest since studying deep tissues in reservoir in humans, which can be defined as HIV reservoirs in organs, is difficult for obvious safety and ethical reasons. As you may guess, deep tissues are only accessible when people living with HIV give their body for science. The goal here is to define how much and under which integrity status HIV persists in different parts of the human body during long-term antiretroviral therapies. Thanks to the generous gift of 2 Canadian men living with HIV, we found that both intact and defective HIV persist in many deep tissues, and that identical HIV viruses can be found in different organs. This is an important step to be able to specifically target the tissues in which most of the intact replication-competent virus is found. So why do we need to study anatomical HIV reservoir? We know that antiretroviral therapies, or ART, do not eradicate HIV. The latently infected cells, which are in a non-replicative state that constitute the HIV reservoir, are not affected by ART. However, HIV reservoir studies are done for the vast majority are performed on circulating CD4 T cells. Few studies, such as these 3 published in the last 6 years in a Science Spotlight presentation from Cure 2021, have investigated multiple anatomical compartments other than blood. They all highlighted the fact that understanding where and how HIV persists in deep tissue is a prerequisite for the development of curative strategies. So here what I'm doing is a report of a case study of 2 people living with HIV and under long-term suppressive ART who gave their bodies for the HIV research to the Canadian collaboratory HIV Cure in 2018. Oops. Sorry. We obtained a high number of near-full-length proviral sequences to be able to identify potential anatomical sites of viral rebound caused by the persistent intact HIV genomes. So basically what we've done to perform this detailed reservoir characterization is we collected multiple samples from up to 15 different organs, and these samples were snap frozen for further quantification. Both autopsies were done inside a 24-hour time frame after death. We focus on lymphoid tissues, including various lymph nodes and the spleen, the different compartment of the gut, but also, sample the liver, the lungs, the spinal cord, the brain, and the testis. So the objective was to categorize the potential anatomical reservoir, and we've done this in 2 phases. First, we wanted to quantify the presence of persistent lethally infected cells in the different deep tissues collected postmortem from these 2 ART-treated participants. To do so, we used quantitative PCR on extracted DNA from multiple pieces of the various organs, and this was performed by my colleagues Maria Julia Ruiz and Amélie Pagliusa. Once we established the presence of lethally infected cells in these organs, we also aimed at characterizing the persistent anatomical reservoirs. To do so, we assess the integrity of the proviruses, the clonality, but we can also assess the distribution of the proviral populations that persist in multiple deep tissues during long-term ART. So we had 2 participants. They were men who passed away in 2018, as I mentioned, but they had quite different clinical histories, which are summarized here. You can see that the viral loads are indicated in blue, CD4 counts in red, and CD8 counts in green, from the estimated time of infection up to the time of death. The first participant died in Ottawa at the age of 67. He was diagnosed with AIDS-related symptoms in 1987. Throughout his life, he was under various ART regimens, all represented at the top of the graph, but had undetectable viral load for the last 11 years. He reported taking his medication until one day before receiving medical assistance at death. He had neither AIDS-related disease at the time of death nor other illness. The second participant died of non-Hodgkin B-cell lymphoma at the age of 68 in Edmonton. Of note, he did not receive chemotherapy for his cancer. He was diagnosed seropositive in 2003, but there is no clinical data recorded for the first 4 years of infection. He was under 2 ART regimen and had undetectable viral load from 2010 to time of death. He suffered from multiple comorbidities, including HCV infection, HIV-associated peripheral neuropathy, and minor neurocognitive disorder. So we started by quantifying total HIV DNA in the multiple deep tissues from multiple pieces. Each dot here represents the mean value of triplicate quantification of a small sample of tissue. Empty dots represent the limit of detection of samples with undetectable values, and a total number of positive samples per tissue are indicated at the top. As for all figures in this presentation, each tissue is color-coded. So what we saw is basically we detected HIV in all tissue analyzed, which represent 15 different tissues for the Ottawa participant and 14 different tissues for the Edmonton participant. But as you can see, the levels measured were variable. we observed that the highest HIV DNA levels are found in the different lymph nodes, all in different blue shades for the Ottawa participant, and in the liver in orange and the spleen in green for the Edmonton participant. Once that we knew that latently infected cells are present in all collected tissues, we then analyzed the genotype of the persistent proviruses in these anatomical sites. To characterize genotypically the HIV reservoir, we did limiting dilution of extracted DNA based on the qPCR quantification. We then amplified near-full-length proviruses using a nested PCR adapted from the FLIPS assay, covering 92% of the HIV genomes from the end of the 3' LTR to the end of the 3' LTR. Every long-range HIV amplicons were barcoded and sent for PacBio next-generation sequencing, a technology that does not require reconstruction steps after sequencing. And in the end, we evaluated integrity and clonality of a total of 441 near-full-length proviruses. So what did we found by characterizing the persistent tissue reservoir? For the first participant, we successfully sequenced 300 near-full-length HIV genomes, ranging from 150 to 9,064 base pairs. They are all represented here in the alignment, color-coded per tissue. These viral genomes were retrieved from 14 different tissues, all indicated at the left, and the number of tissues, the proviruses per tissues are indicated ranging from 1 to 67. We then evaluated the integrity of these proviral genomes. We already know from previous studies that intact, potentially replication-competent proviruses are rare in circulating CD4 T cells in people under suppressive ART, but we know little about the integrity of the anatomical reservoirs. For this participant, only 2% of HIV genomes in deep tissues are intact. These 5 proviruses are located in the spleen, the mediastinal lymph node, and the mesenteric lymph node. On the other hand, 41% of proviruses harbor large internal deletion, 38% were defective due to the presence of hypermutations caused by ApoBec, and 14% presented defects in the Psi locus. The high proportion of hypermutated HIV genomes could be reflecting the period of uncontrolled viremia during the 90s in these participants, since ApoBec is acting during active replication. Overall, as observed here on the Sankey diagram, there is no specific integrity profile associated to a given tissue. For the second participant, we obtained 141 proviral near-full-length HIV genomes, ranging from 490 to 9,051 base pairs. These proviral sequences were amplified from 8 different deep tissues, encompassing 6 to 33 proviruses per tissue. Interestingly, when we investigated the anatomical proviral integrity of this participant, it was a little bit different. Up to 26% of the persistent tissue proviruses are intact. These HIV genomes are detected in the mediastinal lymph node, the jejunum, the colon, the liver, the lungs, and the spleen. The majority of defective proviral sequences contains large internal deletion, the other defects only representing a minimal proportion of all proviruses in deep tissues. Overall, we demonstrated that intact HIV genomes are in minority in deep tissues and are mainly found in lymphoid organs. However, they can also be retrieved in other deep tissues, such as the lung, the liver, and the gut. We then look at the proportion of clonally expanded persistent proviruses. Many studies have highlighted that clonal expansion of CD4 T cell is an important mechanism for the persistence of viral reservoir during ART. Since we sequenced near full-length HIV genomes from ART-treated participants, and knowing the higher rate of the reverse transcriptase, we considered proviruses that are 100% identical to be originating from a clonal expansion of one infected cell. We found that in both participants, clonal expansions represent roughly 50% of the anatomical reservoir. Clonal expansions were composed from 2 to 23 copies. We also observed clonally expanded genomes in every deep tissues where more than one provirus was sequenced, representing between 12 to 77% of the reservoir in a given tissue. Knowing this, if the same proviral sequence is coming from a single cell that has expanded, we can use this as a marker for the distribution of infected cells throughout tissues. This means that a clonal expansion found in only one tissue corresponds to an expanded infected cell that did not recirculate in the body. Here, the size of the pie chart corresponds to the number of different clones found in the different tissues. Each slice of the pie chart represents the proportion of clonal proviruses that are shared with another color-coded tissue, and an empty slice indicates local clonal expansions. So we found that in both participants, each tissue share identical proviruses with other anatomical sites, and that many of these shared clones are also expanded locally. To simplify this, clonal expansion from a given tissue are shared from 3 to 11 of the 13 different organs in the Ottawa participant, and with 2 to 5 of the different 7 organs in the Edmonton participant. We then decided to link every HIV clonal expansion shared between 2 organs to represent the connectivity network between the deep tissues. So basically here, 1 shared clones represent 1 connection. We observed from 1 to 11 different connections between 2 given tissues in the Ottawa participants, and from 1 to 2 for the Edmonton participants. However, what's interesting is that there is no trend demonstrating that connections are clustered between biologically linked tissues, such as lymphoids, organs, or the gut in both participants. This result suggests a lack of compartmentalization of the anatomical reservoirs during long-term suppressive ART. And finally, we observed a significant positive correlation between the size of the clonal expansion and the number of different tissues where these copies are located. So basically, this simply means that a larger clone has more chances to be found in multiple locations. So to summarize this, we demonstrated that latently infected cells during long-term ART persist in all the tissue analyzed in both participants, but at different frequencies. The majority of HIV proviral sequences in deep tissue harbor defects, preventing them to be replication competent, as it is observed in circulating CD4 T cells. Anatomical reservoirs harbor few intact proviruses, mainly but not exclusively found in lymphoid tissues, and half of the persistent reservoir encompasses clonally expanded proviruses that are frequently found in multiple organs. In conclusion, in this study, we performed near full-length proviral sequencing of various human deep tissues, allowing for precise genotypic characterization of the anatomical reservoirs during long-term ART. This project, which supports the last GIFT study results, highlights the presence of persistent HIV in all collected tissues. The 2 participants have different infection and ART histories, a factor that explains the distinct proviral integrity profile. And finally, our results suggest that clonal expansion is an important mechanism of persistence in HIV tissue reservoirs, and that infected cells circulate throughout the body, throughout different anatomical sites without indication of compartmentalization, and also highlight the importance of doing more and more end-of-life research. So obviously, this is a great collaborative CAN-Cure project across Canada, so I have many people to thank. First, I would like to thank my colleagues who participated directly to this project, especially Julia, Amélie, and Rémi from Nicolas Chaumon's lab. A special thank you to Corentin from the CRCHU for the bioinformatic parts. Our collaborating team from Université de Montréal, UQAM, McGill University, Ottawa Hospital, and University of Alberta. The funding organization, of course, the pre-conference organizers, because it's kind to be able to present these kind of results to all of you. Eventually, a big thank you to all of you for the presentation, but my dearest thank you are really to the 2 study participants for their generous gift for HIV research.
Thank you so much, Caroline, for this beautiful tour de force. It is my absolute honor to present today Jeff Taylor. I should really say Dr. Jeff Taylor, who's a 40-year HIV survivor. And for 35 years, he's been an HIV research advocate, starting with the first AZT trials in the late 1980s. Jeff is a community advisor to multiple research networks, including the ACTG AIDS Clinical Trials Group, the University of California, San Diego Antiviral Research Center, the US HIV Guidelines Panel, and the AIDS Malignancy Consortium, as well as co-investigator of the last GIVE study at the University of California, San Diego. Dr. Jeff founded the HIV and Aging Research Project in Palm Springs, which is the community engagement partner for the Reversing Immune Deficiency, or RID, Martin Delaney Collaboratory for HIV Cure. And Jeff will present some of the highlights from the last GIVE study. Thank you.
Great. Thank you, Corinne, for those kind words. And just a disclosure, I have no financial disclosures, but I am not a doctor, so don't get any ideas. So I don't need to tell this audience that the HIV reservoirs is the key problem, and how to clear that is, let me make sure I can get this to, here we go. Okay. Now that we have tech issues. Clearing the HIV reservoir is the key question in HIV. Obviously, the anatomic sites and solid tissues are the challenge. We can measure peripheral blood easily, but it's not just there. And it is the source of viral rebound when we stop ART. So the challenge, of course, is finding those reservoirs. In living people, it can be very difficult to get to those anatomic sites, and even harder to get enough tissue and cells to be useful. And we can't really ethically ask people living with HIV to stop meds when they're doing well on them just for the sake of research. Now, throughout the course of the pandemic, there have been a number of autopsy cohorts that have been established that have proven valuable, but many of these don't have good characterization of the participants prior to death, and very rarely are the autopsies done quickly enough to provide the quality of cells that we need for the kind of research that we're doing now as we start looking for a cure. So the Last Gift study team proposed a solution whereby we approach people living with HIV who were terminally ill with 6 months or less to live, and relying on their altruism to participate in a study that would allow them to do two things. First, to collect clinical data and blood while they're alive, and then upon death, to do a rapid autopsy within 6 hours, which is quite a challenge, to get high quality tissues so we can preserve the quality of the RNA and proteins and able to study them in the way that we need to do cure research. So, as kind of a curveball to the team, there were some questions raised about the ethics and particularly the vulnerability, as they put it, of people at end of life, people living with HIV at end of life, and whether or not it was ethical to ask them to do this, and it was perhaps even exploitive to do so. And so we had to kind of take a pause and think about how we would address those issues. And it really elevated the proposed socio-behavioral research that Dr. Dubé would be leading in the team, and also the role of the community advisors to be able to address these issues adequately so that we could proceed with the trial and convince them that it was going to be ethical and worth doing. So, as we heard earlier in the panel discussion this morning, the HIV population has a long history of participating in research from the very beginning. We wouldn't be where we are today without the incredible altruistic contributions of people living with HIV and enrolling in those first very dangerous trials. They were not always well designed back then. They had a lot of placebo arms and things like that. But nevertheless, people understood the importance and were willing to step up. And that remains true today. And the same group of people also face an incredible amount of death and dying. Many of them came close to death themselves. And even if they did not, certainly had to witness and help their loved ones and families and community members go through that process. So as a community writ large, providers included, as Dr. Sturman described, we have a lot of experience with that. So we're not a vulnerable population. In fact, if there's any population best suited for this kind of research, it is this group of people. And so, under Dr. Dubé's leadership, we did a series of focus groups and reaching out to community groups, the Community Advisory Board, the community where I live in Palm Springs, which I like to call God's waiting room for older people living with HIV, who are that population. And we heard loud and clear that people not only wanted to do this research, but they found it not just patronizing, but infantilizing to be told that they couldn't do this, to take that away from them, because it really is, as we've heard often, a legacy that people can leave. And this population in particular has often been, even though they participated in the early research, has it for decades now, because of their age, comorbidities, multi-drug experience and so forth, been excluded from clinical trials. So it's a real opportunity for them that they were adamant that they be allowed to have, to be able to participate in this kind of research at the end of life. So this resulted in a number of ethical considerations, papers that came out looking at this and describing those details. So from the very beginning, it really elevated the socio-behavioral research aspect of this study. So needless to say, the study went forward. We started in 2017 with Dr. Davey Smith leading, and his co-PI is Dr. Sarah Gianella, who I hope is listening in virtually, and she can hopefully answer any questions that people might have about the biomedical aspects of the study, as I'll be focusing on the socio-behavioral aspects in keeping with the theme for this morning's talks. So obviously the goal is to characterize the HIV reservoirs in blood and anatomic tissues and determine the dynamics of how it rebounds after ART interruption. And the goal was to enroll 5 participants per year. We enrolled 19 in the first phase of the study. So naturally, it's the participants who are the core of the study. They are why we were able to do this, and we were really fortunate to have some phenomenal individuals be our first participants. And I get a little verklempt when I think about these guys, because they were just incredible, both for the research, for the science that they provided, and for the experience that they provided for the study team as they tried to work through all the complex logistical and ethical and psychosocial issues that are part of this research. So the first gentleman, Tony, is shown here with his partner, Blake, and his sister while he was in hospice. And the next one, Max, as you can see, was a very fun-loving individual, was a lot of fun, worked in Hollywood over the years, had a lot of connections, and even though he was afflicted with ALS, had a lust for life, and it was just a delightful person to work with. And so, of course, it takes a village to do this kind of research. And because this is an autopsy study, a critical part is the rapid autopsy team. And as you can imagine, the logistics of performing a rapid autopsy within 6 hours of death is amazing. And they had to kind of work through all those protocols. And also, again, with the assistance of Dr. Dubé, think about the psychosocial impacts of doing this. Often parts of the team are working very closely with the participants before they die, become very close to them, and then they have to, perform the autopsy. So we did a lot of research around that to make sure that, everybody's psychosocial aspects were being considered throughout the team. And then I can't overemphasize the importance of, the study coordinator. In our case, it was a wonderful woman named Susanna. I shouldn't say was, she's still with us. She hasn't died, but she recently retired. So we're feeling her loss terribly. Susanna Concha Garcia. And we were fortunate that she's had decades of experience working with other autopsy cohorts over the years. So she was really well-versed in how to, talk to patients about end-of-life decisions, incorporating their family members and next of kin to support those decisions, and then applied that incredible experience and as we move forward with the last gift study. So if I have any advice for people considering this kind of research, not only do we need to figure out how to cure HIV, we need to learn how to clone people like Susanna, because every team needs not just one, but several of them, because as we've learned, they do retire. People don't stay there forever. So this has resulted in a lot of papers that have come out of this, the research that we've done. As I mentioned, we did a lot of testimonials of the people who were in the study. And this is a common theme: my death will not be in vain. The last gift title of the study is very intentional. People really do see this as a legacy that they're able to provide one last time after basically a lifetime of living with HIV. And also the next of kin, obviously. You can't do this without their support, both for the participants and so that they understand what's happening and don't have any misgivings, during or after the trial. And, another title, of course, is it's now my responsibility to fulfill that wish, as I mentioned, for the autopsy team and what it's like for them, not just the logistics of doing so, but, the psychosocial impacts of participating in that. And then we also made it an effort to not just, publish in scientific journals, but to go to the general media. And you can see the article on the lower right featuring Max and really kind of telling the story of what it was like for him to do this and how important it is, so we can get this out into the general media and to the community, so people understand that this research is happening, how important it is, and that there are opportunities for people living with HIV to provide this kind of support to the cure research endeavor. So, of course, we're looking at the reservoir, and our first paper really did address those primary aims, demonstrating that HIV does persist in deep tissues from multiple anatomic sources throughout the body, which we're uniquely situated to talk about in this kind of study. And I won't dwell on all the lessons learned. There've been so many, and I really, again, want to be focusing on the socio-behavioral aspects. But we've learned a lot about, where proviral DNA exists in different cortical regions, as shown here, both in tissues and in CNS regions. There's a lot of CNS compartmentalization and with the BNABs and brain tissue, and shown by this very elegant figure here. And there's a lot of migration within the brain and across the blood-brain barrier by HIV DNA sequences, as we suspected, again shown here. So there've been, again, coming back to the socio-behavioral aspects, a lot of lessons learned. First and foremost, that the research of end of life provides our participants with a degree of autonomy and agency, and you can utilize their altruism to leave a meaningful scientific legacy to cure a disease that has taken away so much from them, not just from them, but from their communities and the world at large. And, we've really demonstrated that community engagement and making this kind of research participant-centered is particularly crucial in this kind of research. You can't do it without starting with that focus. And I really credit the team for doing that from the outset. And I think it's really contributed to our success and is why others want to emulate what we're doing elsewhere. And as I said, it takes a village, and these kind of multidisciplinary teams are key. And we need to really elevate the role of socio-behavioral research within cure research, because it's so important, as we've been hearing all morning. And all too often, it's an afterthought and does not get funded. It's not included in the original funding. And the same for, including community members. People can't do everything for free indefinitely. So I think it's really important for starting at the beginning when these grants are written and the RFAs go out, that this is a part of it, and how this gets funded is part of the whole process. And lastly, obviously, the decisions regarding the care, both medical care and hospice care, and increasingly medical aid in dying, which is an option in many places like it is in California, are first and foremost to the patient. And the study serves to further the study should serve the interest of the participants and not the other way around. So where do we go from here? There are some next steps as we move into version 2.0 of the study. Obviously, we want to continue what we've been doing and increase the community stakeholder engagement in more diverse settings and more diverse populations. For the interest of time, I didn't include the demographics of the study, but they reflect the demographics of the San Diego HIV epidemic, and that it's overwhelmingly male, mostly white, some Latino, and smaller proportions of African Americans and Asian Pacific Islanders. So it's, obviously the HIV pandemic worldwide is much more diverse, and we need to expand that. And hopefully with collaborations with other groups, we just heard about Cure, and with IAVI in South Africa and India, especially, will provide that kind of diversity, and also looking at this in less resource limited settings. And then also with the ICSTEM cohort in Europe. So I wanted to leave, since we're here in Canada, with the paper that we recently submitted on older people living with HIV, their willingness to participate in end-of-life research in Canada. And it includes many of the people in the room here today. So we're excited to have initiated that kind of collaboration on the socio-behavioral side of the research that's happening. And we're really excited to see that move forward. So with that, I'd like to thank you and thank the organizers for this opportunity.
Thank you very much, Jeff. We'd like to ask our speakers from this session to join us for a panel discussion, and we're going to have about 20 minutes, I think, here you go. We'll have about 20 minutes for this this session. So at this time, we'd like to take your questions. Oh, we have an online question. Is it this one? Okay. This is for everyone. Was the replication competence of post-mortem reservoirs representative of living organs? And will there be organoid studies for more dynamic analysis? Who'd like to take that? Caroline?
Am I on?
Really?
It's an excellent question. By the time of the autopsy was conducted, we hope that it's represent what the living organs was, so that we hope that it represents what the organ were during life for the integrity and potential replication competency of these viruses. I really don't know for organoid model that would suit this kind of further experiment, but that's an excellent question to dig in.
And we also, I don't know, she's not on the big screen. I'm not sure if I can get her. There she is. There's Jessica is also here as well. We have a question from the floor. Please go ahead.
Hi, this is Naz from University of Alberta, Chris Power Lab. This is a question for Caroline. Were you guys able to characterize brain reservoir and kind of figure out if it is intact?
That's also a good point about this this study. Unfortunately, we weren't able to retrieve any sequences from the brain in both participants. We tried hard. We really tried hard, but the amount of potentially infected cells in brain is really low. And we, when I was showing the quantification data first, it wasn't the brain in both cases that there was the most undetectable values from what we tried to quantify. So if we have to do it again, we will need probably a higher amount of tissue to be able to eventually find sequences and maybe find an intact one in the brain or elsewhere in the CNS.
How much tissue?
I don't know the amount properly we did. That's a good question.
Oh, good. Thank you.
Yeah, thanks everyone for fabulous talks. My question's for Carolyn and maybe Jeff, and I'm sure you get asked this a bit, but how do you control for blood contamination of your tissues, given, especially what Carolyn, you showed there was so much interconnection and no compartmentalizations, and tissues like liver are so heavily, blood rich. So how do you control for blood contamination?
If Dr. Gianella is on, perhaps she can address that since she's been involved with the autopsy process.
And in our case, of course, we cannot totally eliminate the fact that there can be a minimal contamination from blood in the tissues, but obviously all the tissues were washed before sampling. So that's probably the best we could have done at that moment. If anyone know a better way to do it, to be sure that there's no blood contamination at all, please come to me and we will write it down. But yeah, that's still something that we can consider, but seeing it everywhere might be less like a lower chance that it's just contamination.
Yeah, because I wondered whether you could use transcriptomics to give you a profile of the degree of mononuclear cells in the tissues that you're actually looking at some stage. It might be important.
So Dr. Genoa made 3 comments in the chat. We plan to formally test this in our cohort and perform some antemortem biopsies to compare. The last GIF is doing a lot of brain work. We have sequences from brain, also sorted microglia. And with regards to the control for blood, we wash the tissues on site and compare sequences with those collected antemortem in blood.
We do have more questions for that, but I actually have a question for Dr. Prasad. I'm very curious, as a woman who's raised a child living with HIV and who had to administer medications and attend a lot of clinic appointments and so on and so forth. I'm wondering, you showed us you had a lot of clinic appointments. I'm wondering if you could share with us some of the challenges of bringing women and their infants into the clinic, and maybe some of the challenges that the women expressed or the mothers expressed about administering treatment, as well as just their own psychosocial needs, and perhaps the expense and burden of that number of appointments. Could you talk to us about that a little bit?
That's actually a great question. And I wonder, are there any participants in the trial, women or no? So most of our studies are, as I said, done in sub-Saharan Africa, and I don't have the direct experience in sub-Saharan Africa, but I will say from our US experience, which is very different than in the African setting, women are very committed to this charge. And and we get asked this question a lot. It is a huge challenge for them to come to clinic. But when we look at our adherence to visits, it's greater than 90%. We have actually very few missed visits. We do have issues around change in family members living with the participants that would lead to some participants withdrawing because of issues around disclosure. Not everyone knows that a certain participant is infected. So it's actually a very complex situation and raises the point that we need to collect more information on participation in these studies and what it means. But the pediatric HIV space, beginning with prevention efforts with as you are well aware of, use of AZT for prevention of mother-to-child transmission, it attests to the commitment of women to have their children lead healthy lives. And so we have tremendous success with retention in clinical trials.
I really wanted to ask that question and also pose that question to Jessica too about bringing women into trials and doing women-only trials, because oftentimes we hear, okay, I'll say it, the excuse that it's too difficult to get women to come to trials. They have too many obligations or commitments and don't participate fully. So I'm hoping that these two women will enlighten you when you're enrolling in your trials to see that you can enroll women and you should enroll women in your trials. Jessica?
So my experience is in the context of Uganda, and certainly we have in some ways an easier time enrolling women than men. It's common for women to bring their children if they're young to the clinic. It's like a welcoming space for women and children. And the men often, frequently, because there aren't as many social supports, get time off work, and there can still be the gender imbalance or the sex imbalance in those roles in the household. So for anyone who's looking to do, research in women, I don't think you're going to face that specific problem working in sub-Saharan Africa. Maybe other issues, but.
Thank you. Nicolas?
Yeah, and my question is for Jessica as well. So you can turn your mic on, Jessica. I was quite fascinated by the data you showed showing differences in the size of the reservoir between men and women, which I think is very consistent with what Eileen Scully and John Corn and Steve Deeks and others have reported in the past. So it seems, as you said, that the reservoir in women might be less inducible than in men, possibly. If that were the case, you would probably expect upon heart interruption, women to rebound later than men. So do we have any evidence that this is the case? I know this is probably very difficult to show, but do we have any data from the literature suggesting that the delay to rebound differs between men and women?
That's a great question. I don't know the answer to it. I'll certainly look for it. I'm not hugely optimistic because of the very low representation of females in cure research that there would be enough women to be able to stratify data to look at that. But I'm going to look for it. I'm going to search for it now. Thank you.
Thank you.
Thank you. We have two related questions. One from an anonymous attendee. What plans, if any, are being planned to collaborate to establish the last give study sites in other parts of the country in order to expand racial, ethnic, gender diversity of study volunteers? And what opportunities and challenges do you anticipate with doing so? And then one question from Moses Supercharger. Jeff, does your work have a challenge of funding, and what plans for the solution? What exact plans to include Africa?
I'll answer the second question first to Moses, and thank you for joining us today, Moses. We really wish you could have been here. It's not the same without you. But yeah, the challenges. Can you repeat his question again?
What plans do you have to include Africa and challenges of funding?
So yeah, for funding, as we move along with this study, they're starting to include funding for community advisors in the grants and in the studies as they move forward. So that's thanks to the study team that have really recognized and elevated the importance of community advisor roles in that. So that's beginning to happen. As for plans for Africa, I know they would love to, and then perhaps Dr. Gianella can weigh in on that, and also other related to the first question, plans for other US collaborations with more diverse populations. 'Cause I know they've been getting lots of requests and a lot of interest, but of course, how they make that happen is another issue.
Certainly, that's a question that often comes up from our Cure Community Advisory Board. And I'd also like to acknowledge that my co-chair of that advisory board, Ron Roseness, he's not with us here today, but he has joined us online, and thank him for his decades of work in HIV activism.
Hi, I have a question. I'm Petronella Ancuta from University of Montreal. I have a question for you, Dr. Perso. Congratulations for your work. I think it's very interesting to see how we can, control the viral replication in these kids for a while with all these advances in treatment. Did you observe any improvement in their immune competence with controlling viral replication? How these kids are born with major defects in immune competence. So do you have any data in this sense?
Yeah, so I'll repeat the question. It has to do with immune competence in children with in utero infections. And to date, if we use the benchmark of immune responses to routine childhood immunizations, we know that with early therapy, we preserve that response. And so that's a key part of staying healthy for children, is actually treating early to maintain immune competence to respond normally to vaccines. Thank you for the question.
Great, thank you. Any other questions? If not, we have a few announcements.
Directly outside,
Great.
immediately.
Okay, so the first announcement is that there will be a group photo immediately outside before lunch. There's lunch in boxes for all the in-person attendees. And we ask that you be careful with COVID-related restrictions, that you avoid gathering inside. And we want to thank all the speakers in the audience for an excellent morning, and we hope you will join us at 2 o'clock this afternoon. Thank you so much.
I think it was great.
Yeah, great job.